Τι σας φέρνει στο μυαλό η λέξη κοκτέιλ? Σε αυτή την ερώτηση οι περισσότεροι μάλλον θα ανακαλέσουν κάτι διασκεδαστικό. Ένα ποτό, μια έξοδο με φίλους, κάποιο παλιό ξέχνιαστο καλοκαίρι σε έναν κόσμο χωρίς πανδημία. Όχι όμως και ο Christos Kyratsous, ο αντιπρόεδρος των τεχνολογίων έρευνας λιμωδών νοσημάτων και ιογενών φορέων στο Regeneron, που φιλοξενείται απόψε στην πλατφόρμα του Athens Science Festival, αναφέρεται σε ένα κοκτέιλ διαφορετικό από τα συνηθισμένα. Μια μίξη που μπορεί να προσφέρει ελπίδα στη μάχη κατά της πανδημίας COVID-19. Στη συναρπαστική του ομιλία με τίτλο «Ένα κοκτέιλ στη μάχη κατά της πανδημίας», ο διακεκριμένος μικροβιολόγος θα μας περιγράψει την προσπάθεια των επιστημόνων για τη δημιουργία ενός κοκτέιλ ανθρώπινων αντισωμάτων που στόχο έχουν την πρόληψη και την θεραπεία της νόσου COVID-19. Επιπλέον, θα μοιραστεί μαζί μας τα ενθαρρυντικά αποτελέσματα των πρώτων κλινικών δοκιμών. Ας πιούμε σε αυτό. Τη συζήτηση θα συντονίσει ο Christos Παναγιωτίδης, καθηγητής μοριακής κυταρικής βιολογίας στο τμήμα φαρμακευτικής του Αριστοτελείου Πανεπιστημίου Θεσσαλονίκης.
Καλησπέρα σας. Με μεγάλη χαρά και ιδιαίτερη περηφάνεια παρουσιάζω σήμερα τον Christos Kyratsous, που θα μας αναλύσει, όπως ακούσατε, τις επιτυχημένες προσπάθειές του στην αντιμετώπιση της πανδημίας COVID-19 με το κοκτέιλ των μονοκλονικών αντισωμάτων που ανέπτυξε. Η περηφάνειά μου έχει να κάνει με το γεγονός ότι ο Dr. Kyratsous, ο Christos, είναι απόφυτος τμήματος φαρμακευτικής του Αριστοτελείου Πανεπιστημίου Θεσσαλονίκης, στο οποίο υπηρετώ, αλλά και απόφυτος στο εργαστηρίο μου. Μετά τις σπουδές του στη φαρμακευτική, ο Dr. Kyratsous συνέχισε τις μεταπτυχιακές και τακτορικές σπουδές στο Πανεπιστήμιο Κολούμπια της Νέας Υόρκης και δούλεψε υπό την επίβλεψη του πολύ γνωστού μοριακού ιολόγου Dr. Saul Silverstein. Για την πρωτοποριακή του έρευνα και τις συνολικές ακαδημαϊκές του επιδόσεις, ο Dr. Kyratsous τιμήθηκε με το βραβείο του Κοσμήτωρα της Ιατρικής Σχολής του Πανεπιστημίου Κολούμπια και μετά το δακτορικό του βέβαια συνέχισε την επιστημονική του πορεία στο εργαστήριο του Μισελ Παγάνο στο Ινστιτούτο Ερευνών Καρκίνου της Νέας Υόρκης, στο NYU, το Πανεπιστήμιο της Νέας Υόρκης. Από εκεί είχε την ευκαιρία να τον αδράξει η εταιρεία βιοτεχνολογίας Regeneron περίπου πριν από 10 χρόνια. Η εντυπωσιακή του πορεία στη Regeneron, όπου, όπως ακούσατε, εξελίχθηκε σε αντιπρόεδρο έρευνας μολυσματικών νόσων και τεχνολογιών υλικών φορέων, δεν μας εξέπληξε καθόλου, τουλάχιστον εμάς όσο το γνωρίζαμε. Πέρα όμως από την εξέλιξή του, τις πολλαπλές επιστημονικές δημοσιεύσεις και τα πολλά βραβεία που έχει λάβει, αυτό που ξεχωρίζει τον Dr. Kyratsous είναι ότι τα αποτελέσματα της έρευνάς του έχουν οδηγήσει σε θεραπείες που σώζουν ζωές, που έχουν σώσει ήδη πάμπολες ζωές. Αυτό δεν είναι κάτι το οποίο μπορούμε να το πούμε όλοι, ελάχιστοι επιστήμονες μπορούν να το πούν. Αυτό έγινε γιατί ανέπτυξε καινοτόμες και αποτελεσματικές θεραπείες για την καταπολέμηση αρκετών ιογενών ασθενειών, όπως αυτές προκαλούνται από τον ιό MERS, από τον αιμορραγικό πυρετό έμπολα και πρόσφατα από τον ιό των SARS-CoV-2. Τα παραπάνω λοιπόν κάνουν ιδιαίτερα σημαντική τη σημερινή του ομιλία, στην οποία θα μας παρουσιάσει κάποια από τα μυστικά του στην ανάπτυξη αποτελεσματικών και ασφαλών κοκτέιλ μονοκλονικών αντισωμάτων για την αντιμετώπιση ιογενών ασθενειών. Χρήστο, συγγνώμη που σου πήρα τόσο από το χρόνο σου. Αλλά, λοιπόν, η ομιλία θα γίνει στα αγγλικά, αλλά τις ερωτήσεις θα τις απαντήσει στα ελληνικά.
Καλησπέρα στην Ελλάδα, καλημέρα από τις Ηνωμένες Πολιτείες. Ευχαριστώ πάρα πολύ για την πρόσκληση, είναι μεγάλη μου χαρά να παρουσιάσω ένα μέρος της δουλειάς μου σήμερα στο συμπόσιό σας και φυσικά να μιλήσω για τα κοκτέιλ αντισωμάτων. Παρενθετικά να πω ότι όλοι μας όταν ακούμε για κοκτέιλ, όλοι μας σκεφτόμαστε λίγο πιο ξέχαστες μέρες και ελπίζω ότι τα κοκτέιλ σαν αυτά που θα παρουσιάσω σήμερα να μας φέρουν πιο κοντά σε αυτές τις μέρες. Η ομιλία που θα δώσω σήμερα είναι για τα μονοκλονικά αντισώματα, για τα ανθρώπινα μονοκλονικά αντισώματα που έχουμε αναπτύξει τα τελευταία χρόνια για διάφορες ιογενείς λοιμώξεις, όπως αναφέρθηκε ο Δ. Παναγιωτίδης. Αλλά η έμφαση φυσικά θα δοθεί στις τεχνολογίες και σε περισσότερες λεπτομέρειες για το κοκτέιλ του SARS-CoV-2. Θα μου επιτρέψετε την ομιλία να την κάνω στα αγγλικά, αλλά πολύ ευχαρίστως να μιλήσουμε στα ελληνικά στην συνέχεια. Όπως είπαμε και πριν, ονομάζομαι Christos Kyratsous και είμαι αντιπρόεδρος έρευνας στην Regeneron Pharmaceuticals στην Νέα Υόρκη, στις Ηνωμένες Πολιτείες. Okay, so just a very brief background on the technologies that help us identify and develop all these antibody cocktails, these fully human antibody cocktails. And our capabilities depend on the technologies that we developed over the last 30 years or so. So Regeneron as a company has spent a lot of time and invested a lot of money to develop a suite of proprietary technologies for drug discovery, development, and manufacturing that can also be leveraged to rapidly respond to emerging threats, like the examples we're going to talk about today. Regeneron's end-to-end capabilities, from discovery of these therapeutics all the way to commercialization, is what we call a Velocis suite of technologies. And these are the exact same technologies that have generated 9 FDA-approved medicines to date. It's a repeatable and a reproducible approach that has changed the timelines to develop these drugs from years to months, including the examples we're going to talk about in infectious diseases in the settings of outbreaks like MERS coronavirus, a coronavirus very similarly, very related to SARS-CoV-2, Ebola virus, and now, of course, with SARS-CoV-2. So the drugs that we have chosen to develop are antibodies. Okay, I'm sure that most of you are familiar with antibodies. Antibodies have this Y-shaped structure and are developed by our body to be extremely specific for their target. Our immune system has been developed to encode millions and millions of different antibodies, and the Y-shaped all the way at the top, the antigen binding region of the antibody is extremely specific for its target. Our immune system has developed over many years to develop these antibodies to combat pathogens, either when we are infecting with pathogens or, of course, when we are vaccinated. And as I mentioned before, they are made extremely specific. They are developed and they are selected to be extremely specific against their target. An antibody against influenza does not bind to SARS-CoV-2, and an antibody against SARS-CoV-2 does not bind to influenza. An antibody against a specific region of SARS-CoV-2 has been developed, designed, and selected to bind to that specific region and not to any other side of their target. And that's extremely important because we want our drugs to be specific for their targets. And of course, antibodies are what help us remember, okay, this is exactly what is contributing to this immunological memory so that we can remember the pathogens that we've been immunized or infected in the past. And that's exactly the reason why we don't get reinfected when we are exposed to the same pathogen in the future. And because of these properties, antibodies make for very nice drugs, okay? You can think, usually when we think of drugs, we think of pills, okay, the small molecules like aspirin, ibuprofen, or antibiotics that we take in the form of a tablet or a pill. Antibodies are much larger. And in the diagram here, you can see the size of the aspirin, okay, and all the way on the right, you can see the size of an antibody. They are proteins. They are made of amino acids, okay, so they would normally be digested, okay, in your stomach and in your, so they must be injected, okay, into your body, either under the skin or intravenously, directly into your blood. But because of the specificity of these antibodies, these as drugs, they have fewer side effects. They don't bind to anything off target. They have very predictable pharmacological properties, That's exactly why we like them as drugs. In addition to that, the antibodies can persist in your body for several weeks. When you take a small molecule like aspirin, for example, the drug is metabolized after a few hours, and then you usually have to take multiple doses in the course of the day or over the course of your treatment. The antibodies can stay in circulation for several weeks. A normal antibody has a half-life or in excess of 3 weeks usually. So what we've been doing over the last 30 years, as I mentioned, is develop several technologies that can help us generate, screen, and scale up antibodies, fully human antibodies in the lab, okay, and develop these antibodies as drugs. And here are some of the key technologies we are using. Velocigene or VelociMouse is a technology, is the world's fastest mouse engineering technology that allows us to do from very, very small point mutation in the mouse genome all the way to megabase scale genetic humanization. So huge swaps of the mouse genome, the human genome, and I'm going to show you an example of that, using a highly automated robotic platform. So very, very easily can swap genes around. This is something we developed about 20 years ago now, and I have a link to the paper if you want to see more details. VelociMune is a proprietary mouse that was engineered using the Velocigene technology, in which 6 megabases of the mouse genome that is encoding for the genes, that is encoding for antibodies, so for the variable regions of either the heavy chain and the light chain of the antibody, have been replaced in the same locus with the human counterparts. And again, I'm going to show you a cartoon of that in a few slides. So basically, these are mice, okay, that where the antibody locus has been fully humanized. And when you are giving a challenge to these mice, and you can immunize them with your favorite antigen, they basically develop fully human antibodies in the mouse. This is in vivo selection of fully human antibodies. And by doing that, you can do in vivo selection of fully human antibody in an animal model. So you don't have to seek either humans that have been previously infected with these pathogens. The VelociMune technology has been coupled with the VelociMap technology, which is an adaptation of a B-cell isolation method. So we are basically extracting the B-cells of interest from these mice, the B-cells that are encoding for an antibody, that are expressing an antibody on the surface that is specific for the target. We are getting these B-cells out of these mice, and we can clone the heavy chain and the light chain of the antibodies into a Chinese hamster ovary cell line, a CHO cell line, that is basically a production cell line that can grow in a bioreactor. It can make very large amounts of these antibodies when you want to create enough material for human trials. And we can do that very, very, very quickly, in as little as 2 weeks. So putting all these things together is what we call the Regeneron rapid response, which is basically a process that utilizes the Regeneron Veloci suite of technologies to rapidly generate, evaluate, and develop fully human antibodies against emerging viruses. So as a recap, why do we want to use antibodies as a rapid response capability? As I told you, antibodies are extremely specific, and very rarely they have off-target toxicity. So the tox profile of an antibody is very predictable, okay, because mostly it's going to be dependent on the binding of the antibody to its target, and you can select for off-screen activity. Unlike vaccines, antibodies can provide immediate protection that can last for an extended duration of time. When you get vaccinated with something, and we're going to go into a little bit more details about that, your immune system needs to react. And that usually takes several weeks. Sometimes you need extra doses in order to generate antibodies at enough concentration to mediate protection. With antibodies, with monoclonal antibodies, the moment you are administering an antibody to somebody, you are immediately reaching the serum concentration that is required for protection, and this serum concentration slowly declines, but it can be way above what is required for protection for a very long period of time. I'm going to show you that it's very straightforward to identify multiple antibodies that are binding different epitopes of the pathogen to prevent from mutation escape. I will show you data why this is important. And of course, with the technology I'm describing today, I'm going to show you how we have bypassed traditional bottlenecks in fully human antibody development and can do this whole process very, 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 very quickly. So just like a recap, antibodies versus vaccines. So obviously, I think in order to combat a pandemic now, like the COVID-19 pandemic, we need a long-term solution that is a vaccine, like vaccines, which are going to provide active immunity. You are giving somebody an immunogen, either virus or a component of the virus, or RNA that is encoding for pieces of the virus, like what is used for SARS-CoV-2 now. The immune system is recognizing those pieces, and then eventually you are making antibodies against these species, those fragments, those antigens. Obviously, these vaccines can only be given for prevention, right? You cannot treat somebody with this kind of vaccines. And immunity is delayed, but is usually long-lasting. Antibodies is a complementary approach that is called passive immunity. So you don't have to wait for somebody to generate immunity, but you are pre-selecting the components that are mediating immunity, and you are passively administering these components to those individuals. You're selecting for these antibodies that are very, very potent in combating those viruses. In the case of SARS-CoV-2, you can purify them, concentrate them, and give them to somebody, either to protect them from the infection for prevention or to treat them after they got infected with SARS-CoV-2. Immunity is provided immediately. You don't have to wait for your immune system to kick in, and it can last up to a few months. It is not as long lasting as a vaccine. So we see those two approaches as complementary, where antibodies is a short-term solution that can also be used for treatment, whereas vaccines is going to provide the long-term solution that society needs in order to combat the pandemic. The processes that I've described you before to generate those antibodies against infectious agents and develop them in the clinic has been done several times now. We have used this Regeneron rapid response for global good for three different applications today. One was against Ebola virus. We developed these antibodies during the West Africa Ebola epidemic in 2014. We selected for a triple antibody cocktail that we called Regeneron EB3. And in a WHO run clinical trial starting in 2018, we saw that the Regeneron EB3 was dramatically superior at preventing Ebola deaths compared to the control arms in this study. Actually, this is the reason why the study was halted, because the Ebola cocktail was statistically superior during an interim read of the data. And because of that, our antibody cocktail was the first FDA-approved therapy against Ebola virus about 6 months ago. Against MERS coronavirus, we generated antibodies, 3048 and 3051, and we worked with NIH to complete phase 1 clinical testing of these antibodies. And of course, against SARS-CoV-2, I'm going to show you a lot of data on that today. We are in the process of developing this dual antibody cocktail in the clinic. And I'm including here 3 publications in Science over the last year, where you can see more details about the work I'm going to describe you over the next few slides. But let me give you a few more slides on the background. As I told you before, the VelocImmune mice are the mice that are encoding for these human antibodies and allow us to generate fully human antibodies in the mouse. Normally, the mouse genome is encoding. You can see here a diagram of the variables in the heavy chain and the variables in the light chain. And basically what we did with the VelocImmune technology, we swapped the mouse regions with the corresponding human regions. So the mouse genome is now encoding for human antibodies. And when you are immunizing these mice, you are getting a hybrid antibody where the business end of the molecule, the variables, are fully human, whereas the constants are fully mouse. And we are doing that so you can retain the signaling of these antibodies in the mouse and things like that. But with a one-step process using robotic cloning, you can basically get those variables and clone them onto your favorite FC. And with a single step, you can have a fully human antibody out of the mouse. As I told you, we are doing this with a VelocImmune technology, where either getting B cells from convalescent humans or splenocytes from the VelocImmune mouse, you can sort for the antigen-positive B cells. You can select the ones that you want, and you can very, very easily convert them into fully human antibodies. And then you can put the heavy chain and the light chain of the antibody into what we call a production cell line. Okay, a production cell line meaning a Chinese hamster ovary cell line that can grow into a bioreactor. The important thing here is that we are making these cell lines immediately after we screen for the antibodies. So the same cell line that is giving the material that's going to be screened initially in vitro and in vivo, so is going to be the exact same cell line that is going to go into a bioreactor much, much later in the process to generate the material that's going to be used in human trials. So the whole process is very, very consistent. You don't have to swap from one cell line to the other that might affect the properties of the antibodies you are screening for. You're using the exact same cell line and a very, 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 very similar material from the very beginning to the very end. Just to give you a visual of how we are scaling up these antibodies, very, very early on, we are using these fairly small bioreactors as a bench scale development in our labs in Tarrytown, New York. Eventually, when we are selecting leads and we want to do toxicology studies, you can see in the middle these larger bioreactors, also in our facility in Tarrytown, New York, that can generate several grams of material that can be used for these animal studies. And then eventually, when you have something that you want to develop as a drug, we are transferring those cell lines into our manufacturing facilities, either in Rensselaer, New York, or in Raheen in Ireland. And these tanks are sometimes 10,000 liters or 20,000 liters bioreactors. So they are gigantic steel tanks, and you can generate kilograms of material with every run of the bioreactor, which runs for about a couple of weeks. So having said all that, let me provide you some details and share the data with you on how we screened for the antibodies that are now the 2-antibody cocktail against SARS-CoV-2. And I'm going to share some of the early clinical data we are getting with these antibodies. SARS-CoV-2, as you all know, stands for Severe Acute Respiratory Syndrome Coronavirus 2. It's an RNA virus, and is the causative agent of the COVID-19 pandemic. When you look at the virus, it has a single glycoprotein on the surface that is called spike. It's a trimer that you can see here in this reddish color. And this is the protein that is required for the interaction of the virus with a host receptor called ACE2. Okay, so in order for the virus to get inside the cell, this interaction between spike and ACE2 is required. The virus binds to ACE2, and that's exactly what's going to cause the fusion of the virus to get inside susceptible cells and initiate a round of replication. Of course, the first cases were reported in China more than a year ago now, and the WHO declared the pandemic on March 11th of 2020. So pretty much at the same time, we started our response, to generate antibodies against this virus. And at the top here, I have a rough timeline of our process. So we immunized our mice on February 5th of last year. The immunized were generating an immune response over the month of February. We started isolating antibodies at the beginning of March. And we also got a sample from 3 humans that have previously infected and recovered from COVID-19 disease a few weeks later. So for the second half of March, as well as the first half of April, we were basically screened human antibodies that were coming out of the 3 humans, and human antibodies that were coming out of the velocimune mouse. And we were comparing and contrasting the properties of these antibodies in order to select for the best antibodies that we go into clinical development. What we wanted to get is 2 antibodies that are potent neutralizers, potent blockers of the virus, get breadth against all known variants of the spike receptor binding domain. And I'm going to show you why we were looking for a cocktail, for these antibodies. At the time, we screened about 3,300 antibodies. Of course, this is a process that since then we've done multiple times. To date, we have screened more than 25,000 antibodies against SARS-CoV-2. And we screened all these antibodies in multiple different assays, including in vitro neutralization assays to see how well they block the virus, how well they bind to the virus, how tight they're binding to their target, if the Fc domain can signal to effector cells and things like that. And using all these assays, we ended up with 9 different antibodies that can mix and match in 17 different combinations that were evaluated for their neutralization potency as cocktails. This diagram here is showing you the ability of these antibodies to block the virus in vitro. So you see here that as you are increasing the concentration of your antibody, you are increasing their ability to neutralize, which means their ability to block the virus to enter inside susceptible cells. And that is true no matter if you are using a VSV, a vesicular stomatitis virus that has been pseudotyped with SARS-CoV-2, or if you are using the actual virus. Increasing amounts of the antibody can block the virus. And back then, what we wanted to do is we wanted to make sure that these antibodies are binding to regions of the virus that do not change very often. As you very well know, viruses are sloppy. Every single time they replicate, they make mistakes, and the replication machinery makes mistakes. And so as they spread in the human population, these mistakes end up accumulating, and sometimes you are selecting for variants, either because these variants are spreading easier in the human population, or because they can evade immune pressure if you have previously infected with the virus, okay? And that's exactly what we see today. The variants are becoming something that you very commonly hear and read about in the press. But of course, this is something that we started screening for back in April of last year, when the number of infections was much lower, because we were anticipating that this is going to become an issue in the future. So back then, what we did is we analyzed a library of variants that was available at the time. You can see here that we analyzed 22,000 variants, and we made every single receptor binding domain mutant in the lab so that we can screen all of them. We can screen the ability of the antibodies to bind to all of them, and you can see the substitutions here on the left. To date, there are about 700,000 of SARS-CoV-2 sequences that have been deposited in multiple different databases, and we are still making every single one of them, every single separate variant in the receptor binding domain, and evaluating every single one of them in the lab, because we want, of course, our antibodies to retain their activity as the virus evolves in the field. And then we started looking for antibodies that can bind simultaneously, okay, on the surface of the virus. And here is a biochemical assay. We don't have to go into the details, but basically what you're looking for is a binding signal here. A number greater than 0 is indicating signal. And that number is the biochar, the binding signal of the second antibody, when this first antibody is already binding on the target. So when you have an antibody binding on the target and the second antibody wants to bind to the same epitope, you're basically getting a signal that is close to 0. For example, when you do this assay with one antibody and you are trying to bind the second antibody on top. When you have an antibody that is binding on one side and a second antibody that can bind to a distinct site, then this is a non-competing pair of antibodies, because they can bind independently of each other. For example, Regeneron 33 that you can see here, and Regeneron 87, okay, is a non-competing pair of antibodies. And that's why you can see this fairly large binding signal of the second antibody when the first antibody is pre-bound to your target. So this is a biochemical assay that can help us identify pairs of antibodies that make sense and can bind simultaneously and independent of each other, because that's exactly what we want when we have a drug. You don't want your drug to be composed of two components that are competing with each other. You want two antibodies that can bind independently. These findings were then validated using structural work. Here you can see the efforts of our cryo-EM group that can basically show, this is in blue here, you can see the receptor binding domain of the spike protein. And you can see here the heavy and the light chain of one antibody. And here you can see the heavy and the light chain of the other antibody. And you can see how, exactly as predicted by our biochemical assays, they can bind independently on the surface of the receptor binding domain. Okay, so we now have the structural confirmation that our biochemical assays were accurate. But why does that matter? Okay, why do we want cocktails of non-competing antibodies? Why is this important? The reason is what I told you before. Viruses change. Okay, so when you're putting pressure on them, when you are treating a virus with a single drug, then the viruses are going to try to evade and escape the pressure that you are applying on the growth of the virus. And that's very, very common in the virus literature. We've known that for multiple years. That's why when you are treating HIV infections, for example, you never treat with one drug. You treat with cocktails of 3, sometimes even more drugs, because you want to apply multiple different pressures on the evolution of the virus. So this is the experiment that shows that. What we are doing here is we are taking virus and we are incubating the virus with decreasing concentrations of the antibody. As expected, in large concentrations of the antibody, you see no growth. But in small concentrations, in low concentrations of the antibody, you see what we call cytopathic effect. So the cells start dying because the virus cannot replicate. If you take the virus from an intermediate antibody concentration and you passage that over and over and over again, eventually what you're going to start seeing is you're going to start to see a shift to the left, because the virus will start being able to grow in higher and higher concentrations of your antibody. So what you see here is that when you are doing this experiment with a single antibodies, and it doesn't matter what antibody that is, you're very, very, very quickly start seeing this shift to the left, because you are selecting for variants that can grow in the presence of these antibodies. When you are treating with a cocktail, competing antibodies, okay, based on the competition assays I showed you before, you have the same thing, because molecularly, although you have two different entities, they are binding to the same target. So a single amino acid change can basically knock out the binding of both antibodies. You lose binding of both antibodies, and then you don't have a drug anymore. However, when you are treating with cocktails of non-competing antibodies or partially competing antibodies, okay, so things that can compete in the biochemical assays, but you have separate epitopes, you can see that you are basically prevent the ability of the virus to escape, because you are multiplying the pressure of these antibodies on the genome of the virus. And I'm going to show you in a few slides why that is important in vivo. So the next thing we wanted to do is find out if these antibodies can work in vivo, in animal models. And what we used in this experiment is rhesus macaques, these monkeys, that can be infected with SARS-CoV-2. Okay, the infection in these monkeys is fairly transient, but allows us, okay, to see if our therapeutics can work in an in vivo setting. So what we are doing is we are giving these antibodies 3 days prior to infection. Then we infect these animals with the virus. And then every day or so, we are either collecting a swab from their nose or we are getting fluid from their lungs. And what you can see here is the placebo animals. The virus goes up initially, and then eventually it starts going down. Okay. If you are treating these animals with our antibody, you can see with red the median of the placebo, whereas down here you see all of the animals. Basically, you are completely preventing any detection of viral RNA in the nasal cavities in these animals. When you're collecting fluid from the lungs, you basically see pretty much the same thing. In the placebo-treated animals, you see a transient viremia, and then eventually the virus goes away. When you're treating with your antibody, you're almost completely blocking the ability of the virus to establish an infection. So we've done this experiment both prophylactically and treatment. We've also used the hamster model of infection, and we showed that these antibodies can work very well in vivo. And because of that, we started a fairly complex clinical development program. And we wanted to test if this antibody cocktail can be used in the clinic to either prevent or treat SARS-CoV-2 infection. So we ran 3 different trials. For pre-infection, for prophylaxis, we are looking at high-risk exposure. So what we are doing is we are doing what we call a household contact study. So if you have a household, a family, and one member of the family is detected positive for SARS-CoV-2, then you're giving these antibodies to the rest of the household because they live together. So they are at high risk of being exposed to the virus and getting infected. And you are looking at preventing basically a transmission of the virus to the rest of the household. For post-infection, we are doing 2 different trials. One is an outpatient trial, which is early treatment. Somebody that is positive for the virus, has symptoms of the disease, okay, but it doesn't need to be hospitalized yet. You give them the antibodies, and then what you are trying to prevent is hospitalization, and then of course death. And then eventually we are also doing, a hospitalization trial, where we are giving these antibodies to people that are already hospitalized because they have severe symptoms of SARS-CoV-2 infection. And there are different cohorts there, depending on their oxygen requirement. And the endpoint here is either lack of progression, okay, to higher and higher flow oxygen or ventilation, and of course, death. And we are running this trial both here in the US and also in the UK as part of the recovery trial run by the NHS, the healthcare system in the UK. The difference is that the UK trial is only using mortality as their endpoint. So what we've learned, okay, during these trials, first of all, is the very important role of endogenous antibodies. Now, as I'm going to show you, having antibodies against SARS-CoV-2 or being able to develop these antibodies very, very, very quickly is exactly what's going to prevent progression of the disease to more severe, okay? And I think the data is very, very clear. When you are giving our monoclonal antibody in trans, okay, to people that are, especially to the people that are lacking these endogenous antibody responses, then you are substituting basically for these, for the lack of the endogenous antibodies, and you can prevent progression of the disease. And I'm going to show you all the settings where that applies. So first of all, let me share some of the natural history data here. And this is some data both from the outpatient, the early treatment trial, and also the hospitalized patient trial. So here there's no treatment. Everybody is being given the placebo, okay? But we are looking at the role of antibodies, okay, at the role that antibodies have on disease. So as you can see here, when you are measuring viral load in this outpatient setting, the people that at the time of enrollment do not have their own antibodies yet. They're seronegative. They haven't had time to develop their own antibodies. They have a very high viral load. The people that have already developed their antibodies, their virus is about 1,000 times lower. It's about 3 logs lower, okay? So when you start developing your symptoms, if your immune system has already kicked in and has developed their own antibodies, your viral load is already much lower, okay? And then you can clear the virus, okay? And you can bring it below the level of quantification much faster than if you don't have your own antibodies. Now, if you take these two populations and you look at their progression, okay, if they're going to need medical attention, you see that the people that have their own antibodies, they don't really go to the doctor. Their symptoms are resolved by themselves, okay? They have mild disease. They don't need to go into the ER. They don't need to be administered into the hospital. The people that don't have their own antibodies, these are the guys that are at high risk, okay? These are the guys that end up going into the ER, and these are the guys that end up being hospitalized, and some of them end up dying, okay? Clearly demonstrating how our own antibodies can prevent can greatly affect the outcome here. And the same thing happens in the hospitalized setting. So some people, when they are hospitalized, they have their own antibodies. Some people, they do not have their own antibodies. Some people do have antibodies. And you can see there's about a 2-log, 100 times difference in the viral load at the time when they go into the hospital. The people that do not have their own antibodies are the ones that end up requiring a ventilator, or the ones end up dying. But the ones that have their own antibodies, they end up doing much, much better than the people that do not have their own antibodies. So the question is, if you take these seronegatives and you give them the Regeneron antibody cocktail, what happens? So as you can see here, when you look at the viral load, you are basically converting the seronegatives into the seropositives, because you can start clearing the virus much, much faster, okay? You're giving them an antiviral, something that blocks virus replication, and you end up accelerating the clearance of the virus in their body, both in the outpatient trial and in the hospitalization setting. You don't have an effect in the seropositives. Why? Because these people already have their own antibodies. So giving them more antibodies doesn't really make a huge difference. But is that important? Do you change the outcome? And the answer is yes. So this is in the outpatient trial. This is the phase 2 data. And you can see that in the placebo-treated patients, you are slowly accumulating events, okay, throughout their trial for at least 2 weeks. However, in the people that are getting the antibodies, for the first 5 days, they pretty much mirror the placebo, but then you flatten the line, okay? So 5 days after people get these antibodies, they basically do not require medical attention. They're not going to prevent and become hospitalized, and eventually we have no deaths, okay, in this arm of the study. And in the hospitalized setting, you see pretty much the same trend, when the placebos are slowly accumulating events, okay, which is progression into ventilation and death. Whereas the treated patients, again, over the first few weeks or so, they are tracking the placebo, but then you're also almost flattening the line, okay, because you are stopping virus replication faster, you are clearing the virus faster, and eventually these people end up doing better. Now, so this is the data that I showed you before is in the post-exposure treatment setting, either early treatment or late treatment. In the post-exposure prophylactic setting, in the household setting, what we've shown in our phase 2 part of the trial is that we are basically blocking symptomatic PCR in the household by 100%. Okay, so nobody, okay, that was treated with the antibody okay, even when they got infected, developed any symptoms. So you can basically prevent transmission of disease in the household. And out of the ones that were the high virus PCR positive, we saw 100% reduction in infections. Again, showing that these antibodies can work very well as antivirals. And only 5 days ago, we published the phase 3 component of these studies that basically mirror the result of the phase 2 studies in a much larger setting. So we now have the larger clinical trial of its kind. We basically validated all the data. So I hope that what you've seen now is that there is strong scientific rationale for utilizing antibodies to treat SARS-CoV-2 infection. We have a pair of non-competing antibodies that can protect against variants. And you can see benefit from that in multiple different clinical settings. And we are showing an association between mounting an endogenous response, especially an antibody response, and the requirement or the benefit that you are getting from the administration of these exogenous antibodies. And what I want to end up with is a few more slides that are showing how we can stay ahead of the virus, and especially when it comes to variants and escape, especially because the variants are such a popular topic now because of all the news that and the stories that the variants are getting. And again, what I want to highlight here is the rationale for selecting for a non-competing pair of antibodies. So here you're seeing the neutralization curves of the individual antibodies, okay, 33 and 87, as well as in dark blue, the antibody cocktail. And you can see the neutralization curves against multiple different recently emerging variants of this virus, okay? So this is something that emerged last summer and now has taken over the entire planet. This is the UK variant that started being detected just before Christmas. You can see the South Africa and the Brazil variant that were pretty much came up at the same time, the California variant, and the 484 that is very, very prevalent here in New York. And what you can see here that even in extreme settings, like the South Africa or the Brazil variant, where one of the antibodies loses, okay, some of the ability to neutralize, the other antibody is still there and is doing the job. And that's exactly, again, another reason why you need your two antibodies to bind to two completely separate sites of the virus, because you can afford your antibody to lose one of the antibodies, to lose its ability to neutralize. Your cocktail is still functional. If you were out with monotherapy, just like many of our competitors are, and you had an antibody that was binding to an epitope very, very similar to antibody 33, then you basically don't have any activity against any of these variants, okay? You lose the activity. And the last thing I want to show you, okay, is how we are preventing the escape in vivo. I showed you some data before, how a cocktail of antibodies, unlike the monotherapy, can prevent the escape in vitro, but how does that translate in an in vivo setting? So this is a hamster study, okay, where the animals are either treated with monotherapy, 33 and 87, either with a placebo control or with the antibody cocktail. What we are doing here is we are treating these animals, and a week later, we are doing a deep sequencing, okay, of the virus in the lungs of these animals to try and detect any variant, okay, that's going to come up. In this pink, okay, whatever the color is, orange, pink color here, you can see the receptor binding domain of the spike protein of the virus. This is the site where the antibodies are binding. So when you are treating the animals with placebo, you don't really see much variability because there's no pressure. When you are treating the animals with Regeneron 33, then you start picking up these two mutations at the receptor binding domain. This is exactly where the epitope of 33 is. When you treat these animals with 87, again, you are applying pressure and you start seeing variants that are within the epitope of 87, exactly as you expect, exactly as what we see in our in vitro studies. However, when you are treating the animals with a cocktail, okay, then you don't see any variants at all, okay? Again, because if something like that, okay, tries to come up in the population, then your second antibody is there to suppress that virus population. If something like this, okay, comes up in the virus population, then the other antibody is there to suppress that viral population. And the end result is that you don't see any variant that is coming up. And finally, that's exactly also what we see in our clinical studies. Here you can see some deep sequencing data from about almost 5,000 samples from all the participants in the outpatient and the hospitalized setting. Everybody from the phase 2 was sequenced, or the virus from everybody in the phase 2 was sequenced. And basically what you see here is some random variants that are accumulating, and both the rate of accumulation as well as the sites are not on the antibody epitopes, okay, and are also the rate is exactly the same between placebo and the 2 treatment groups, okay? Again, indicating that the cocktail can prevent the emergence of this escape in vivo. That's exactly why we believe that cocktail therapy is exactly what should be used in the clinic. And with that, I would like to thank everybody who participated, okay, in the work to select and develop these antibodies. Here you can see some of the names of the people that participated in the preclinical efforts, okay, to characterize these antibodies. There is an equal or even larger number of people that participated in the clinical development program, okay, for these antibodies. It truly takes a village to develop a drug. There are hundreds and hundreds of people that are involved. And of course, I want to thank you for listening to this presentation this afternoon. And I'm happy to take any questions. Thank you very much.
Χρήστο, σε ευχαριστούμε πάρα πολύ για μια πάρα πολύ καλή ομιλία, μια υπέροχη ομιλία και επίσης για τα πολύ ενθαρρυντικά αποτελέσματά σου που μοιράστηκες σε σχέση με την αποτελεσματικότητα του κοκτέιλ των αντισωμάτων. Βέβαια, όπως όλοι γνωρίζουμε για τα κοκτέιλ, υπάρχουν διάφορα γούστα. Λοιπόν, επομένως, είναι πολύ πιθανόν ότι να χρειαστούμε κι άλλο κοκτέιλ. Εάν τα παραλλάγματα συνεχίζουν να εμφανίζονται με την ταχύτητα που εμφανίζονται, που για κάποιους είναι παράξενοι, ίσως να χρειαστούμε κάποια καινούργια κοκτέιλ. Θα ήθελα να σε ρωτήσω, πόσο γρήγορα περιμένεις ότι θα γίνει η ανάπτυξη ενός καινούργιου μείγματος αντισωμάτων, αν χρειαστεί, και πόσο χρόνο θα πάρει αυτή η διαδικασία όσον αφορά την αδειοδότηση. Γιατί δύο ζητήματα. Ένα είναι να το φτιάξει, δεύτερο είναι να πάρει το emergency use approval. Λοιπόν.
Αυτό που έχουμε κάνει, έχουμε δουλέψει αρκετά, όπως είπα, τα αποτελέσματα που έχω δείξει τώρα είναι με το κοκτέιλ που επιλέξαμε μετά από το screen περίπου 3.300 αντισωμάτων. Αυτή τη στιγμή έχουμε δεκάδες χιλιάδες αντισώματα τα οποία έχουμε περάσει από την αντίστοιχη διαδικασία και έχουμε διάφορα άρα κοκτέιλ ή πιθανά κοκτέιλ τα οποία μπορούν να χρησιμοποιηθούν για οποιοδήποτε από τα variants. Το καλό είναι ότι τα variants ναι μεν εμφανίζονται, αλλά βλέπουμε συγκεκριμένα patterns. Για παράδειγμα, το 484 είναι ένα αμινοξύ το οποίο φαίνεται να είναι ένας από τους κύριους στόχους της ανοσολογικής αντίδρασης των ανθρώπων. Οπότε το βλέπουμε στην Νότια Αφρική, το βλέπουμε στη Βραζιλία, το βλέπουμε στην Ευρώπη και το βλέπουμε και σε αρκετές πολιτείες στις Ηνωμένες Πολιτείες. Οπότε αυτό είναι υπάρχουν μερικά αμινοξέα τα οποία είναι ίδια σε όλο τον κόσμο. Οπότε αυτά μας δείχνουν ότι μερικά sites στον ιό είναι πιο εύκολο να αλλάξουν και επίσης υπάρχει και η πίεση για να αλλάξουν. Οπότε αυτά είναι τα sites που προσπαθούμε εμείς τώρα αυτή τη στιγμή να αποφύγουμε για όλα αυτά τα μελλοντικά κοκτέιλ. Αυτό που θέλω να πω είναι ότι τα αντισώματα υπάρχουν ήδη για όλα αυτά τα μεταλλάγματα που υπάρχουν, που κυκλοφορούν ήδη. Επειδή υπάρχουν οι κυταρικές σειρές, μπορούμε μέσα σε ένα διάστημα 2 μηνών περίπου να έχουμε το φάρμακο το οποίο είναι έτοιμο για τις κλινικές δοκιμές. Αλλά φυσικά, όπως αναφέρατε, υπάρχει η συζήτηση με τα regulatory agencies, τόσο στο FDA στις Ηνωμένες Πολιτείες, όσο και το EMA στην Ευρώπη. Αυτό που θέλω να θυμίσω είναι ότι για τα εμβόλια, για παράδειγμα, για υγειικές λοιμώξεις, όπως είπαμε για το εμβόλιο της γρίπης, τα components του εμβολίου αλλάζουν κάθε χρόνο. Είναι παρόμοια, αλλά λιγάκι διαφορετικά. Έχουν την ίδια βάση ουσιαστικά, αλλά είναι ένα διαφορετικό στέλεχος του ιού της γρίπης. Οπότε δεν χρειάζεται να κάνεις κλινικές δοκιμές για κάθε εμβόλιο κάθε χρόνο. επειδή ουσιαστικά το φάρμακό σου δεν αλλάζει 100%. Είναι πιθανόν κάτι τέτοιο να γίνει και με τα αντισώματα. Γιατί ο στόχος των αντισωμάτων δεν αλλάζει, είναι το receptor binding domain του ιού. Ο τρόπος με τον οποίο δουλεύουν είναι ο ίδιος, είναι neutralizing antibodies εναντίον του ιού. Οπότε είναι πολύ πιθανόν να περάσουμε σε ένα μοντέλο, όπως ακριβώς τα εμβόλια, που να μπορούμε να κάνουμε mix and matching των αντισωμάτων, αναλόγως με το ποιο variant κυκλοφορεί στον κόσμο.
Πολύ ωραία. Και κάτι τελευταίο, γιατί ο χρόνος πιέζει λίγο, αν και λέει θα είναι καλό να κλείσουμε, όσον αφορά τη χορήγηση. Λοιπόν, μέχρι τώρα η χορήγηση είναι νοσοκομιακά. Αναγκαστικά με ενδοφλέβεια έγχυση. Υπάρχει κάποια περίπτωση που να γίνεται από τον ασθενή.
Βεβαίως. Την προηγούμενη εβδομάδα, στα Phase 3 αποτελέσματα που δείξαμε την προηγούμενη εβδομάδα, δείχνουμε ότι τόσο η ενδοφλέβεια έγχυση όσο και η υποδερμική χορήγηση των αντισωμάτων δίνει ακριβώς τα ίδια αποτελέσματα. Οπότε είμαστε σε ένα μοντέλο τώρα που μεταφερόμαστε από την ενδοφλέβεια έγχυση στην υποδερμική. Και αυτή μπορεί να γίνεται είτε από τον ίδιο ασθενή, είτε σε οποιοδήποτε γραφείο του γιατρού, χωρίς να χρειάζεται η ενδοφλέβεια έγχυση. Οπότε έχουμε πολύ ενθαρρυντικά αποτελέσματα και από αυτό.
Πολύ ενθαρρυντικό. Ευχαριστούμε πάρα πολύ τον δόκτωρα Kyratsous και μέχρι την επόμενη ομιλία, λοιπόν, σε χαιρετούμε. Και ευχαριστούμε και το Athens Science Festival για την ευκαιρία.